Hello, my dear students. So, in the previous class, we discussed about cell cycle regulation. Today's class is about cell cycle checkpoint. Myself, Dr. Shibu from the Department of Biotechnology. Let's move to the topic. So, we all familiarized with what is meant by cell cycle and what is the regulation of cell cycle, and also we discussed something about checkpoints. Okay. So, what is meant by checkpoint is, as I earlier said, how the check post is to prevent the illegal drugs or illegal material to come into the state or there's a border. They protect the illegal items from one area to another area. Similarly, cells also having a checkpoint to monitorize whether the cell is perfectly okay or not before dividing. Okay. So, introduction to the cell cycle checkpoint where the normal cells, however, move through a cell cycle in a regulated way. They use the information about their own internal states and the environment signals around which them they can decide whether to proceed or not. Okay. So this regulation makes sure that the cell don't divide under unfavorable conditions. For instance, if there is a DNA damage or if there is any room for the cells to accommodate, the cells won't divide. Checkpoint is a state in the eukaryotic cell cycle at which the cells can examine all the internal and external cues. Okay, and they will decide whether to move forward or not. Okay, so that is so this thing it is mentioned. Then there are a number of checkpoints, but most important checkpoints is this three. G1 checkpoint, which is present in between this G1 and S transition, then the G2 checkpoint, where in between the G2 and M transition, and there is a spindle checkpoint between metaphase and anaphase. Okay. So checkpoint. So as I said, G1 checkpoint present here in between this G1 and S are having a G1 checkpoint, and in between the G2 and M we are having G2 checkpoint, and we are having a spindle checkpoint in between the M phase, that is in between the metaphase and anaphase. So what is meant by G1 checkpoint is the G1 checkpoint is the main decision point for the cell, that is the primary point at which it must choose whether or not to divide. Once a cell passes the G1 checkpoints and enter the space, it became irreversible and committed to division. So that's what the G1 checkpoint will check. It is the primary checkpoint and will choose whether the cells to divide or not. Once a cell is passes, we already mentioned that it is irreversible process. We cannot able to the cells cannot able to come back. So that is barring unexpected problems such as in the case of DNA damage or replication error, the cells that passes the G1 checkpoint will continue the rest of the way through the cell cycle and produce two daughter cells. If the G1 checkpoint doesn't check the DNA damage properly, what will happen? The cells will be going to produce two daughter cells, and that is having an error. The error will be passing from generation to generation. Okay. So at this G1 checkpoint, the cell checks whether the internal and external conditions are right to divide. What is the G1 checkpoint checks is that it will check the internal and external condition and the signals whether it is okay or it is right for the division. There are some of the factors that cells will access. Okay, or the first one is size, then the nutrients, then the molecular signal, then DNA integrity. Size, it is mentioned, is the size is larger enough to divide, then the nutrient, does the cells have enough energy reserves are available to divide. Then molecular signal, is the cell receive positive signal from the neighbors, and the final one is DNA integrity. Yes, it is the DNA damage or not. Okay, this is what checked in G1 checkpoint. The second checkpoint is G2 checkpoint, where I already mentioned that to make sure the cell division goes smoothly and to produce a healthy daughter cell without any undamaged DNA, the cells has an additional checkpoint before the stem phase, that is called as a G2 checkpoint. At the stage, the cells will again check the DNA integrity, any DNA damage or done, and DNA replication. Was the DNA completely copied during the S phase or not? If any error or a damage are detected, the cells will pause at the G2 checkpoint and allow for it repair. If the cell repair mechanism or the checkpoint find out that detecting the problem with the DNA, the cell cycle is halted. The cell cycle is halted, and the cells attempt to either complete DNA replication or repair the damaged DNA. If the damage is irreparable, in case if the cells cannot able to repair it, what it have to do with this, the cell may undergo a process called as apoptosis, one of the important process that is called as apoptosis or programmed cell death. Okay, detailed study of this apoptosis and programmed cell death we'll discuss later because we have this in our syllabus. So this self-destruction mechanism ensure that DNA damage is not passes onto daughter cell, and it is important in preventing the cancer. So in such cases, if the DNA damage is irreparable, the cells may undergo a process called as apoptosis or called as programmed cell death or cell suicide. We'll go for a suicide or cell destruction mechanism, ensuring that the damaged DNA will not pass from one generation to another. Instead of giving a false result or a false DNA to the another generation, the cells will commit suicide. Okay, to avoid the cancer. So that is the G2 checkpoint. The third one is the spindle checkpoint, where the M checkpoint, otherwise called as M checkpoint, also called as spindle checkpoint. Here the cells examine whether all the cysteine chromatids are attached to the spindle microtubules. This is happening during the phase called as anaphase. You know what is meant by anaphase. During the anaphase, what will happen? The cell separation or the chromatic separation will be happening. All the chromosomes are attached to the spindle fibers. Okay, because of the separation of cysteine chromatid during the anaphase is an irreversible step, as I already said. Anaphase is once the chromosome is separated, we cannot able to join it back. It is an irreversible step. So the cycle will not proceed until all the chromosomes are firmly attached at least two spindle fibers from the opposite pole of the cell. Okay, so the cells will wait if the chromosomes are attached to all the spindle fibers, because we know how many chromosomes is there. Whatever chromosome we are having, all the chromosomes should be attached by the spindle fiber. How this cell checkpoint will work is, it seems that the cells don't actually scan the metaphase plate to confirm that all the chromosomes are there. Okay, how it will work here is, it doesn't check for the metaphase plate to confirm that whether all the chromosome are not, because anyway all the chromosomes should be there. Instead, what it will do, they look for a straggler chromosome that are in a wrong place or are floating around in the cytoplasm. Okay, so what is the role during the anaphases? All the chromosomes should be attached by the spindle fiber at both the end. Both the end it should be attached. So if it is attached by both the end, it will happen firmly attached in the meta region. If any spindle fiber is not attached properly, you know that you can see the straggler chromosome. It will be hanging around and floating in the cytoplasm. So the cells will look for only the straggler chromosome and they will find out. If the chromosome is misplaced, the cells will pause at mitosis, allow the time for the spindle fiber to catch the chromosome. If it is found that some of the chromosome is not attached by the spindle fiber, will wait. The cells will wait. It will not go for proceed in the next stage. It will wait for the things and allow the spindle fiber to catch the chromosome. And after that only, it will be moving forward. Okay, so that is called as a spindle checkpoint. Hope you will understand about the checkpoint. So we mentioned about three major checkpoints: G1, G2, and spindle checkpoints. Okay, if you have any doubts, you can contact me. So that's all today. See you in the next class.